Hello, and welcome to an abbreviated overview of DNA replication. We'll start simple. This is a prokaryotic cell. This is what its DNA might look like, and let's assume it's a circle. And sometimes it just wants to divide as fast as it can. The DNA is usually anchored to the cell membrane, and two replisomes start working. But then, if we take a step around, the newly replicated DNA also gets anchored to the membrane, and the cell elongates, pulling the two strands apart. Before the cells are separate, replication begins yet again, and so on. Now let's take a look at some of the finer details. This is an origin site. It's where everything begins. This actually occurs in a massive protein complex called the initiation complex, but for the sake of not getting confused, we'll only go over the ones essential to replication. Some proteins, like topoisomerase and helicase, open up the DNA into a replication bubble, which is composed of two replication forks extending in opposite directions. It would be nice to know where our 3' and 5' ends are so that we can see the polymerases orient themselves correctly. They can only assemble new nucleotides onto the 3' end of the strand. There's also a certain symmetry here, courtesy of the DNA's antiparallel feature. Can you see it? Okay, well, that's enough of that. For now, let's follow the left fork as it continues zooming off and... oh, well, that's funny. It seems to have stopped. What could be happening here? Well, if we get a little closer, we'll find that the DNA has become much too twisted for the replisome to continue. So, what now? Luckily for us, this is topoisomerase, and it is tasked with unwinding the DNA. As we continue to push up the strand while straightening it out behind us, the front just keeps getting twistier. At this point, the enzyme breaks the phosphodiester bond, lets the whole thing spin around a bit, and then reseals the cut. Good as new. Helicase breaks the hydrogen bonds keeping the bases together, resulting in two strands. Single-stranded binding proteins then attach to ensure that they don't immediately come back together, or at least not before new complementary DNA has had a chance to be synthesized. Speaking of which, here comes DNA polymerase on the leading strand, and it's gone. Now let's take a closer look at that lagging strand, which, aside from its direction and length, is synthesized through exactly the same process as the other one. The resolution's a little low, so maybe if we... that's better. In case you were wondering where the nucleotides came from, well, here they are, just floating around. So, primase, an RNA polymerase, lays down a few nucleotides from NTP monomer units, before it moves on to bigger and better places. Why did we need this? Well, that's because DNA polymerase is lazy and needs everything done for it. But once it has its 3' starting point, and when it's kept in place by the sliding clamp, it can lay down dNTPs across long sequences with startling accuracy. This is due in part to its ability to sense the shape of and detach nucleotides it has improperly paired, which is referred to as 3' to 5' exonuclease activity. This newly synthesized, smallish strand is known as an Okazaki fragment. DNA polymerase replaces the primer, and any missing phosphodiester bonds, like this one, are catalyzed by ligase. And there we go, a full double-stranded DNA molecule, completely indistinguishable, ignoring any inevitable mistakes, from the original, in accordance with what is known as the Semiconservative model of replication. Half of each chromosome consists of newly made DNA, and the other half of previously existing template DNA. And now we can do this all over again. Forever.